Hello, this is Gina Piscatelli with your third lecture on week 4 for Anatomy and Physiology 1. Uh, Today we're going to talk about the first two steps of cell metabolism, and that is glycolysis and the transition reaction. So I'm just going to hit some highlights that we can see in this particular slide, and that is that the first step, glycolysis, is going to involve the breakdown of glucose. It's going to get turned into pyruvate. The second step is going to involve changing pyruvate into something called acetyl-CoA. I tried to depict with this cartoon that glycolysis happens in the cytoplasm, whereas the transition reaction, or step 2, happens in the intermembrane space of the mitochondria. Also noticeable on this diagram are some, I'm going to call them byproducts of the first two steps. Glycolysis produces a net of 2 ATP, and it produces NADH. And the transition reaction produces NADH and carbon dioxide in addition to this acetyl-CoA. So if you recall, the 4 steps of aerobic cellular respiration include glycolysis, the transition reaction, then the Krebs cycle, then the electron transport chain. When we use glucose as a starting material, all 4 steps are used. So we're just going to cover the first 2 today. And also I just wanted to remind you of the location. As I mentioned previously, step 1, glycolysis, happens in the cytoplasm. The transition reaction, or we call it pyruvate oxidation, occurs in the second step, the intermembrane space. And then the Krebs cycle, which is a series of chemical reactions, happens in the matrix of the mitochondria. And the electron transport chain happens at the inner mitochondrial membrane. Since we're going to be doing glycolysis and the transition reaction, I just want to emphasize to you that I'm setting up these lectures so we kind of do one step at a time. If you have not first seen the overview of cell metabolism lecture, you'll want to look at that first. So the process of glycolysis, defined as glucose breakdown, or the lysis of glucose. Now the whole process is 10 chemical reactions that require enzymes. Oxygen is actually not required for this first step. So under anaerobic conditions, cells can perform glycolysis. We start with this glucose, which is our organic reactant, and our organic product is pyruvate. In addition to that, we're going to make some NADH from NAD+. Technically, that's organic as well, right? Because it's a nucleic acid. And of course, in the process, we're making 2 ATP, which are it's a nucleic acid, so that's organic, carbon-based. And I forgot to put the reactant, of course, would be ADP. And then it's going to get turned into ATP. Okay, this is an extremely simplified version or picture of glycolysis, and it's really small. And what we start with here is a 6-carbon glucose. So there's like 6 pearls, 6 circles representing the carbons there. Now to get the process started, 2 ATP are used, broken down. So here's an ATP that gets broken down. What's occurring during this process is that the glucose is becoming phosphorylated. Any enzyme that phosphorylates a compound is called a kinase. So you can bet that the enzyme that does this reaction is going to have kinase in its name somewhere. But ultimately, after phosphorylation, the the glucose carbons get split up so that 3 and 3 are located in each of 2 molecules. Those 3 carbon molecules get transformed with multiple chemical reactions, and in the end we get what are called pyruvates. And the pyruvate is made up of 3 carbons, and I have a picture of its chemical structure in this green box over here. There's 1, 2, 3 carbons. Okay. Now, since bonds are broken, there's free electrons around, and those electrons are going to hop onto NADH. So NADH is produced. Also, the original phosphates that were added to get things started, they will eventually get broken off and used, maybe not those exact ones, but at least replenish the store of phosphate so that in each of these transformations to pyruvate, we make 2 ATP. So the net result is that 4 are made, but we net 2 ATP. So that's just, sorry, I'm trying to write here. It's hard when the mouse is slower than you are. Okay. It's just sort of a summary of what happens. Here are the reactions, and you will not have to draw these out or understand them per se, but this slide shows you the first 6 reactions of glycolysis. And if you take a biochemistry class, you will be required to first know the name of the reactant and the product for each of the 6 steps. So the product of the first step becomes the reactant of the next step, and so on and so forth. In addition, in biochemistry, you got to draw it. You have to draw the structures too. But we aren't going to do that. I'm just showing you that it's a little bit complex. We go from glucose to glucose 6-phosphate because we used an ATP. Then that gets turned into fructose 6-phosphate, then fructose 1,6-bisphosphate, blah, 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 and it keeps going down the line. Okay, so notice that NADH is produced at a certain step. Please also notice that each step requires its own enzyme. Okay, so there are 2 enzymes, better erase that. There are 2 enzymes that will phosphorylate something. So hexokinase does a phosphorylation of glucose, and phosphofructokinase will actually phosphorylate fructose 6-phosphate. Doesn't matter. This is steps 1 through 6. We started with glucose. We've used ATP. We've made some NADH. And then when you get down to steps 7 through 10, now you get to actually make some ATP. So here's a step that makes ATP. Now remember, we are 3 carbon molecules from this point on. So there are 2 series of reactions happening at the same time. We started with 6 carbons, but now we have 2 3 carbon molecules, both going through these processes. We make some more NADH, more ATP, but the bottom line for us is that pyruvate is the end product of glucose breakdown or glycolysis, and it has 3 carbons. Okay? Now, under aerobic conditions, that means when oxygen is around, that pyruvate will cross the outer membrane of the mitochondria, go into the intermembrane space, and be changed. It'll undergo pyruvate oxidation. That's what will happen. So pyruvate oxidation involves losing electrons and ultimately making carbon dioxide. So we're going to look at that just a bit. So pyruvate is our starting material, organic material for the transition reaction, and it is converted into a compound called acetyl coenzyme A. Now acetyl groups have 2 carbons. So that tells me right away I've lost a carbon somewhere. And where it goes is into carbon dioxide. This coenzyme A is kind of a complicated molecule, so we just abbreviate it. Now in this process of being oxidized, meaning it loses electrons, the electrons are going to go to NAD+. So we made NADH, and I'm making a big deal about NADH because it will be, it'll have good big payback. It'll help us make tons of ATP in a cell. So here's the picture of pyruvate with its 3 different carbons. Notice that acetyl-CoA has just 2 carbons, 1, 2. This is that coenzyme group that we're not going to worry about too much. So no ATP production. Okay, so for the transition reaction, for 1 glucose that's got 6 carbons, we actually have 2 pyruvate starting molecules, and they're converted to 2 acetyl-CoA molecules. Therefore, 2 carbon dioxides are made, 2 NADHs are made, but no ATP is produced at all. That's what happens if oxygen is around. Now, what if there isn't any oxygen? So let's talk about that for a second. So this slide looks more complicated than I guess it needs to be. So the question is, what's the destiny of pyruvate? Well, when oxygen is present, pyruvate's going to go to the mitochondria, and what we just saw is what's going to happen. Acetyl-CoA is going to be made. But when oxygen is lacking, this 3-carbon pyruvate gets turned into lactic acid. And lactic acid stays in the cytoplasm. This is also called anaerobic fermentation. Now eventually that lactic acid will change, it will release some gas, it'll release electrons, but it isn't the same process as what we see with ATP production. Now the act of changing pyruvate to lactic acid does give off a little bit of ATP. Okay, a little ATP does come off here, but not nearly as much as we're going to get by sending the pyruvate in the form of acetyl-CoA to the mitochondria. This is where we're going to get a lot of ATP production. Oops. So the question is, after acetyl-CoA has been formed, what occurs? Well, in our cells, because again, we're aerobic organisms, the acetyl-CoA is going to cross into the matrix of the mitochondria and start the process of the Krebs cycle, or step 3, which I'm going to discuss in another lecture. So glycolysis and the transition reaction both produce NADH. These NADHs we don't worry about yet, but those electrons that they're carrying will go to the electron transport chain. I call it the ETC. Okay. Now, only one of the 2 processes actually formed ATP or gave any ATP net production, and that's glycolysis. And the one that neither necessarily require oxygen, but the one that produced carbon dioxide is the transition reaction. Okay? The organic end product, considering the nutrient as our starting reactant, for one of them, the product is called pyruvate. So glucose gets turned into pyruvate. In the transition reaction, pyruvate gets turned into acetyl-CoA. So that's the summary of those two steps. Now it's time for you to watch one on the Krebs cycle and then the electron transport chain. Thank you.